Thank everybody for coming today. Conda, my audio is okay, correct?
Yeah, you're good to go.
All right. Unpause my screen that you guys are looking at. We're back to being live. All righty. So we might as well get going. So my name is Charles Sywin. I'm the program director for the HEAL Non-addictive analgesic therapeutic development program for small molecules and biologics. I will be turning over the reins of this program over to Marianne Palamounter, probably in the by end of September timeframe. She's on the call today as well. We're listening in. Mike Loshinsky is responsible for the overall HEAL pain preclinical portfolio here at NINDS. And Anne-Marie Brome has responsibility for biologics within the Division of Translational Research and assists us in this program for the projects that are biologics based. We hope today to tell you about the Analgesic Therapeutics Development Program, but also give you sort of, Michael is going to give you a broader view of what the whole Therapeutics Development Program looks like from the beginning through the end. And then we'll sort of narrow in on the optimization efforts under this FOA. So have you started recording, Wakanda? I guess is my first question. Because if not, I need to say to everyone else, this will be recording. Yes?
Yes, I have.
Excellent. Okay. So we're part of the HEAL program, the NIH Helping to End Addiction Long-Term Initiative. And we're under the pain research priorities, and we're really trying to enhance the pain management. And that has many facets, including understanding the biological underpinnings of chronic pain, hopefully accelerating the discovery and preclinical development of non-addictive pain treatments, advancing those treatments into the clinical pipeline in order to inform best practices for effective pain management while minimizing the risk of addiction for the future. And there's, if you're interested in all HEAL programs, you can go to the website, go to look at the top of there. There's open funding opportunities. You can see not only this funding opportunity, but all the ones that are open presently. We have programs across the spectrum from discovery all the way through clinical trials and implementation and dissemination. I'll be focusing on my talk about the optimization efforts. Michael will be talking a bit about the planning and initial therapeutics development program, but also note that we have other support programs that are not grant-based, but service-based, and that includes through the preclinical screening platform for pain, as well as NCAT's efforts in this program where they will work with you as a collaborator, and then in the clinical space under EPIC-Net. Overall, we've realigned the programs to really focus on deliverables, focus on the scientific challenges, and minimize the program complexity and handoffs from one to the other, and incentivize industry to hopefully reinvest in this area. So I'm going to turn this over to Michael to go through the Analgesic Development Program and the early stage FOAs that can help fund you. And the purpose of that is to really sort of say, today you may fit into the optimization program, but you may be too early for it and you'll know what's available to you, or you may be too advanced for it and you'll know what's available to you on that side of the equation. So Michael, are you ready?
Thank you, Chuck. Sure.
I'll turn the pages as you asked me to.
Thank you. Next slide, please. So with the HEAL Analgesic Development Program, we've developed a suite of application choices or funding announcement choices for you to come into this program at whatever stage your particular project is at. So take a look at the left side of the screen where it mentions the planning grants. So those are planning grants to get you ready for these large initial therapeutics development grants. That's the U19. So the R61, which is RFA NS 21-029, it's on the left side of the slide there, is a planning grant to help you with team building, getting a good rationale for the mechanism of action or discovery of the target you want to work with, and then showing that you can work together as a team, and that's the feasibility. Now that is a 2-year grant to plan and get you ready for the U19. Now, if you've worked together before and they're aware and they're very good application, they're good publications already for this particular target, it's well validated, then maybe you could come directly into the U19, where there's 5 research components. We'll go through each of those, and some of them are summarized there on the screen. And these are large projects that include multiple individual research projects, all working together to develop a hit to lead, which will get you to the submission requirements for Chuck's UG3, UH3 optimization stage. And then with the IND approval and the phase 1, which can be covered by the UG3, UH3, as Chuck will mention later, then your asset can be ready for our phase 2 EPPIC-Net clinical trials network for pain. Next slide, please. So on the subsequent slides, it says R34. This is a typo, and this is my fault. It should be R61. There was a previous FOA that this is referring to. So these are all R61s, and on the previous slide is the correct number for that. It's NS21-029, right? So the R61 are 2 years, and again, the focus there is team building, building preliminary data for that U19 submission, and the budget is up to $500,000 per year. There's no opportunity for a renewal, okay? The U19s have an entry criteria of having that multidisciplinary team in place, and the expertise that you need is the different types of expertise that are needed for having a pain therapeutics or analgesic development project. It means expertise in pain biology, even the clinical aspect of pain, also assay development, screening, PD, and PK studies, and developing of pharmacodynamic biomarkers. You also need a good rationale for the biology of the target that you're working on, and some good preliminary data, and that's the goal of that planning grant, but it's not required that you apply for the planning grant first. You can come directly into the U19 if you already have that in place. The U19 is up to 5 years, and the direct costs are capped at $1.5 million per year for each of the 5 years, but we don't expect all of the research projects to last the duration of those 5 years. Next slide, please. So for those R61 applications, that multidisciplinary team, what we're referring to is expertise in pain biology, project management, statistical analysis, really good experience with developing rigorous experiments, assay development, and all the other activities that happen in drug discovery, be it small molecule or in biologics. And then the early drug development of the PK studies, just having that expertise in place. You don't have to have all those activities in the R61, but a plan for having that, building that team so that you can apply for the U19. Next slide, please. So the clinical benefit and the potential for this plan of treatment is an important thing to build into even the R61 planning grant stage, okay? And that validation of that target so that you can have a very strong U19 application, this is also very important. The developing your assay or doing initial screening, that can happen in the planning grant stage, but again, if you need more time than that, it's appropriate to finish that up in the U19, okay? It's also important to address the addiction potential of the target you're going after and have plans for determining whether or not your intervention will have little or no liability for addiction. Next slide. So now the U19, I mentioned, have multiple components. Halfway through the slide there, you see this list of 5 different components: the validation of the therapeutic target and the underlying biology, development and validation of the animal models and outcome measures, the assay development, screening, and early optimization, discovery and validation of the pharmacodynamic markers, and the efficacy and PK/PD studies. Now, these components are required. And if you already have done that work, and let's say you already have an assay and a screen done, or let's say you don't need to develop an animal model, you just have to give a rationale of why you're not including that. But you have to have at least 3 of these components, and you have to address all 5 of them. Don't put them out of order, all right? The research components have to be submitted in this order in the application. The data management core and the administrative core are both required, and the research core may or may not be needed. But if you do need it, it should be used for 2 of the research components or more, because otherwise it just looks like an extension of the research project, and that might not do well in review. Okay, so although not a requirement, the research core, if it's going to be a part of only one project, you just roll it in. Next slide, please. So again, for these U19s, you have to have that multidisciplinary team in place and show that they can work together. This is to help you during review. Demonstrate the clinical benefit of that potential pain treatment, and the rationale for that approach. If you don't have it in place, use the R61 to get that rationale. And the overall integrated therapeutics development plan can be for up to 5 years, but not all of the components have to last for that duration of the project. Data management core is required, and that's to interact with the HEAL data ecosystem, and the website that describes the HEAL data ecosystem is on the slide there. And it'll be milestone driven, and we'll have regular meetings to monitor the progress of the project. Next slide.
I think that's it, Michael.
Okay, thanks, Chuck.
All right. So thank you, Michael, for giving sort of the overview of the sort of the FOAs that come before us to get us ready, to get you ready for our program. And so you hear what's available there. So now we'll switch gears and talk about RFAN S21-010, which is the non-addictive analgesic therapeutic development program. It is, again, milestone driven. We hope to identify and conduct all the studies necessary to really get you through the IND enabling studies and onto the clinic in that first in human experience. The end goal is to identify a clinical candidate. We do that by optimizing and doing the IND enabling studies, filing the IND, completing the trials. But there's also a component, and it's an important component, is can you identify a companion target engagement biomarker? You should include that in your application, and you should have a conversation. If it's not possible, why not? Seeking partnerships is obviously an end goal for all of these so that you have the money to carry it further. But we're trying to get you ready for a phase 2 trial, and we recognize you may not be able to do that, and that's why Epicnet exists. The program itself is all, as we said, in this stage, and we support all these activities. The key thing you should know is it's a cooperative agreement, and that cooperative agreement meaning that NIH is going to be actively involved with you. We'll meet with you every 2 weeks probably in what we call the lead discovery teams. We will identify and conduct experiments through the non-disease biology experiments through our CROs, if that's what you've asked for us to do. And we provide access to expert consultants and CROs. And you may also be able to tap into the PSPP or NCAS if you work before your application to make that agreements in place. So like, for instance, NCAS requires you have a CRADA in place before your application to us. So they know exactly what they're agreeing to do as part of the application. We support the IND enabling studies and phase 1, and we can do those through our network, but you could also plan to do these yourselves. So what does our network look like? Presently, our network is still being developed for the full spectrum of small molecules and biologics, but we are leveraging, and we already have an existing network for small molecule space from the Blueprint Neurotherapeutics network that I manage. And we will leverage a lot of the contracts that exist there, as well as putting new contracts in place of similar ilk. Oops, and I realized I didn't get rid of the second AMRI thing here. AMRI's new name is Curia, and we have Curia in both manufacturing as well as in medicinal chemistry. We have a relational database which we can give access to if you're doing optimization work. We have PK tox contracts, and we have clinical contracts that allow us to move things forward through all the different steps. The key element here is that they're all directed through the lead discovery, lead development teams that are, as I said, meeting every couple of weeks. They're really comprised of the principal investigator, you all, and industry consultants that the NIH provides, and NIH staff who are there to help facilitate and support you. You must, in the grant, at a minimum, bring in all the disease biology, bioactivity, efficacy studies, addiction, potential addiction studies as part of the grant application. You can leverage these contracts, and we recommend you talk to us about those so that we can ensure that we can do what you want us to do. And importantly, you can also utilize your own. So if you already have a pre-established relationship, and it'd be like reinventing the wheel to have a synthesis redone by somebody else, then perhaps you can propose to do that yourself with your existing contractors. Talk to us about it. We're not we don't have an objection to doing that. We just want to know from a perspective of what makes the most sense. From a planning point of view, especially if you're doing optimization of things, either through ARC, through the MedCam, or through, we're establishing contracts with the biologics. We're doing optimization efforts. Talk to us about it, because what we don't want you to do is go off and hire people, and then if you don't meet your milestones, you have to get rid of those people if we have contracts for which that risk can be mitigated for you. But if you already have an established team, that's less of an issue. And we do expect you to hit the ground running. So again, if you don't have those people up in place, we can hit the ground running, in our case, by putting up the contracts, but it'll be more difficult perhaps for you. So again, just think through those things. We plan to launch up to 17 projects in the first 3 years of this program, and we're in the process right now of launching our first projects from the earlier receipt dates. Remaining receipt dates for this coming year are October 15th, February 15th of next year, and June 15th. And then we'll have a pause in receipt dates, and we'll have to determine at that point whether we plan to reissue the FOA. The goal is to have at least 5 projects move into the UH3. So that tells you we do plan to have attrition, right? We do plan to hold people to milestones and look carefully on their progress to make sure that we are, in fact, moving the best projects forward. As I said before, we do have to focus in on biomarkers to support target engagement and pharmacodynamics in the early clinical work. So again, we're looking for PD, response, target engagement biomarkers, safety biomarkers, but it's really designed to interpret the IND toxicology and hopefully help project phase 1 human dosing. We're not really talking about doing, patient selection biomarkers for phase 2 and things like that. We're really trying, in that early read, can we get an early read of target engagement? All projects will enter in the UG3 phase, and so that's important to note, right? We do expect most projects will enter at the discovery phase of the program, although it is possible to come in and say, I'm at the development phase. Historically speaking, I have found very few applications have all of their data already generated to say that they really have a development candidate. So usually they're typically at the end of lead optimization with a lot of characterization left to be done in order to determine whether it's a clinical candidate. So again, I advise people to be careful on how you propose your application, because you can box yourself in. If you think, I think I have the development candidate, but I have these other ones, and you just say this is the development candidate, and we're going to push forward on that one, and will be no optimization or no further work, that boxes you into a pretty tight corner if there's something in the characterization of that primary compound goes awry, then the project's over really quickly. So do be careful about this. There's a maximum of 2 years for the UG3 phase, and you can have a UH3 phase of 3 years. Applications must have both the UG3 and UH3 activities in the application and in the budget. So if you have 5 years, I expect 5 individual budgets for each of those years. I do also expect to know what clear go, no-go milestone in the UG3 phase is, preferably at each year of the UG3 phase. So you are expected to lay out a path that goes from the beginning to the clinic, but you don't have, if you're going to use our contracts to do sort of first in human healthy volunteer study, then these won't be clinical trial applications. You will check no to that box because the clinical trial will be done through our contract. If you plan to ask for the money to run the clinical trial yourself, then the application is a clinical trial application, and you have to look through whether, if it's a delayed onset, put that in. If not, put in all the information that's required for clinical trial. Do talk to us, right? Talk to us about your budgets, talk to us about your plans. We can help you in terms of making sure that you're laying it out in a way that makes sense in terms of making milestones on an annual basis, as well as having the correct budget for things. We do have 9 ICs participating in this, along with the Office of Research on Women's Health. It's important to note that all the grants are NINDS held grants, and so the people you see here will help as science officers and disease biology expertise, and they will hopefully attend LDTs as needed to provide additional input to your teams when requested about how you fit into the overall portfolio of in this area, help in the competitive landscape and all of those things there that they have expertise in. So as I said, the overall goal here is to get to phase 1, and the entry criteria is a rigorous biological rationale for the intended approach. It doesn't mean you have to have the target. You can have a phenotypic program. It'll make obviously the target engagement biomarker problem a little more complex, and you may not be able to do that, and you can explain why that is. You also need to have a promising small molecule or biologic starting point for optimization. And you need to have scientifically sound assays to optimize, to drive the structure-activity relationship work or the optimization work in the biologic space in order to identify the better compounds and test the agents on a regular basis. That is what you need to come in. And you need, again, depending on where you're coming in, you should explain in your application what stage you really are at. And don't try to say I'm overly. If you try to oversell where you're at, the reviewers will say, okay, where's the data that supports that? And so it's important sometimes if you're earlier, say, I'm really in the benchmarking stage, I've got compounds, I have these series of things, and what I'm going to do is benchmark these. I want to go out and get the PK, I want to go get the characterization of these molecules done in terms of in vitro ADME profile in order to pick the best series. That's fine. You can say that and just clearly lay it out as your work plan. If you're in the truly in the optimization stage, clearly lay out, here's my SAR, here's my structure property relationships. I need to optimize for X, and I'm going to drive my SAR in this next stage, and the go, no go is going to be related to, can I improve X while maintaining the good properties I have today. If I'm coming in as a development project and I still need to determine whether I have a therapeutic window. It could be, here's my compound and I need to run the dose range finding tox studies so I actually have a reasonable therapeutic index. And that will be my go, no go decision point, whether I move to the next stage. So think about that, talk to us about it. We can really help here. The scope of the program, it supports preclinical optimization and development of small molecules, leading to the assembly and submission of an IND and the implementation of phase 1. The reason why I'm highlighting this is we sometimes get applications where people saying, I have this known molecule from the literature and I want to just run this and go into phase 2. And so that would be out of scope for us, right? Because we're really aimed towards doing the IND and moving to phase 1. If you're doing a surgical procedure with known other agents, again, it's going to be difficult to put it within this program. We do have other programs that potentially could fit into, but make sure you hit these key points. You're doing preclinical optimization and development of a small molecule or biologic. That's got to be the major component of what we're doing. And then it needs to have an end goal of submission of the IND and running a phase 1 clinical trial. So activities that are not responsive to this FOA: screening to identify hits, basic research and studies of disease mechanism, animal model development, development of diagnostic and diagnostic devices. Again, there are other FOAs to do all of that work. Studies directed beyond phase 1 clinical testing, things like aiming for EPPIC-Net, and projects that are really out of the pain area, such as opioid sparing projects and projects targeting mu-opioid, for which we have de-emphasized those, and you could potentially have a conversation with NIDA about them and opioid use disorder. So in the big picture, ensure your application has separate budgets for all 5 years. Like every submission cycle, I'm scrambling to get people to fix this prior to review, so that they can get their budgets reviewed. Clearly indicate in a table, it's requested in the FOA, put a table together that says, here are my assays, here's what I need to get done. We're going to conduct this part ourselves. We want this part done by NIH contractors, and we need this help with consultants. If we not, and are they going to be NIH consultants or are they going to be our own consultants? You can also have sort of a table of proposed activities that includes the throughput and who's going to do it. You can combine both these tables into one table many times if you're clever. And you can also, and it's required to have a target product profile table. And that's simply a table that says, here's my indication, here's my route of administration, here's my ideal, here's my acceptable criteria for a drug to be. Many applications do not address at all. They're silent by the biomarker component. That hurts them in review. I mean, you need to either say, I'm going to develop it or why I'm not going to develop it. If you're asking NIH to conduct the clinical trials, please mark your application as no. Clinical trial, no. If you're planning on doing it, mark it yes. And this is important because if you mark it yes and it's not, and it's not a clinical trial, it still gets reviewed as if it is, and things are missing. So I can't change that marker for you. There is a mistake in the FOA, which was corrected with a notice, which is in the FOA. If you look under the link in the FOA for this notice, that says the matching requirements are not there anymore, right? There was just one spot it didn't get removed, and so it was removed with this notice. So you do not need cost matching for this FOA. Again, phase 1, all projects must enter a UG3. Don't come in with just a UH3. You must have things we discussed in terms of small molecule, rigorous rationale, target engagement. We encourage optimization for potency and efficacy, and having those efficacy studies for pain models is important to pick the appropriate pain models for the indications that you are proposing. It's also appropriate and important to characterize the molecules for ADME properties, especially if you're saying you're in a later stage, like hit to lead or lead optimization. The reviewers are going to want to know what information you have at ADME. If you don't have any of that, you're still in the benchmarking stage. Select and characterize the lead candidates. be ready for, are you really ready for in vivo testing? If you're not, say I'm not, all right? The compounds aren't ready, and say, we need to optimize this in order to have a compound to test in vivo. We're basing our hypothesis on the literature. Here's the preliminary data. It was rigorous data, and that's why we think this is a viable project. In the second phase, there will be an administrative review that determines how we move from UG3 to UH3. And as I said, not all projects will move. But we will base it on things like further optimization that's needed, the likelihood of success there, the development of the biomarkers, the non-GLP tox studies. Do we have the ability to set the GLP tox doses? Can we get high enough exposures to actually run a GLP tox study? PK formulation, cell bank and development and testing, gene expression levels, biodistribution, tumorigenicity, immunogenicity. Can we make the molecules, the process development activities associated with that, manufacturing associated with that. And then ultimately in later stages, in IND enabling safety, genotox, herd toxicology studies that we might have for a development project, those will all be potential things that we would evaluate at the UH3. And it will be dependent on that administrative review that includes staff and leadership, and they will determine if a project is going to advance based on all the things we talked about relative to achieving your milestones, the overall feasibility of that project, including the biomarkers, the competitive landscape, our own internal program priorities, and lastly, the availability of funds, right? And we typically don't have the funds to take everything forward no matter what at risk. So if you're not making your milestones, that does put you at significant risk. Some things to really do here: read the opportunity carefully, discuss the proposal with the program directors here at NIH. We're happy to help. Stick to the page limits. Don't, do put forth a scientific premise and address the rigor in the research strategy section. Don't bury it in the vertebrate animal section. You need to discuss, this is a properly powered, blinded, controlled study in my preliminary data that I've cited, as well as the data I plan on generating. Address obvious criticisms up front. If you have a molecule with a structural alert in it, you better be saying, I plan on getting rid of that quickly. And here's how I plan to do that. If I'm working in tight patent space, you need to sort of explain how you're going to get out of that. If you're dealing with, I need to convert from one biologic sort of method to another biologic method, how am I going to do that rapidly? So you just need to really do that. I've talked about the scientific premise and rigor. Complete your registrations at least 6 to 8 weeks in advance. So if you're new to NIH, make sure you get all this stuff done, and keep in mind things will be changing. We're migrating some of the registration stuff as we go into 2022, so keep an eye on that. Consider submitting your application early. It gives you a chance to react to issues that might arise, and discuss your budget with NIH staff if you have questions. Talk with your tech transfer people. You need to plan for funding, patents, and licensing activities. You can't ask the NIH to do that for you. Some things not to do. Don't plan this to be your sole funding source, because it can end abruptly if you miss your milestones. Don't forget to address the use of NIH contract resources. If you're silent about it, it really confuses the reviewers. And don't plan to use NIH contracts for disease biology, because we just don't have contracts to do that. Don't include our contracts or letters of support from our contractors in your budget if you plan on using us to contract for you. And don't refer to prior review comments. And this is important for anybody coming back in, especially because we only have new applications, resubmissions are not allowed. So if you put in a page of saying I responded to the last reviewer's comments or last review, your application will get withdrawn by CSR before it ever gets reviewed, and there's nothing I can do about that. So please, by submitting early, avoiding these things, you have a chance to address these issues. So this did come up in the last round. So I'm happy to take questions now. Anything from the application process, the review process, the budgets, proposals, contracts, intellectual property. I should mention, and I didn't highlight it as we went through, our contracts for the manufacturing and for the optimization effort, things that would likely produce intellectual property have special clauses in them where we assign the intellectual property to you all, institutions, and our consultants will assign their intellectual property to the institution. And NIH staff are endeavoring not to become inventors. We're there to facilitate. And so the goal here is to keep your intellectual property with your institution so you have unencumbered IP in order to license it down the road. So I'm going to pause my screen here if I can, Rakhanda, so I can open up your chat screen. Rakhanda, you can put it on the other one. Where you plan on putting the information?
On the Skype.
On the Skype, okay. I have Skype set to do not disturb, so I'll have to change that. Give me a second.
Mm-hmm. While you're doing that, Chuck, I'll also mention that this presentation, as well as the recording, will be sent out in a couple of days. It will be featured on our BPN YouTube page. So I will send that link in a couple of days along with the presentation slides.
Okay. All right. So I have your questions up. So at least the first few. So the first one, I'll take the sort of reverse order. We are working on acupuncture to identify biomarkers for pain management. Does this fit this call? It does not, because we talked about the optimization of a small molecule or a biologic, and that would be more of a procedure. And I don't know whether Michael would have any suggestions on where that might fit, or Marianne.
I think if they're looking for biomarkers, Marianne might be a better person to give suggestions. Just for clarification, the question is related to biomarkers, correct?
Correct. Well, it's working with acupuncture and identifying biomarkers is also.
Right. So if you're looking for a mechanism of action or the efficacy of acupuncture, that's not going to respond to any of these forwards that we're talking about here. But if it's a biomarker associated with that therapeutic intervention, maybe Marianne can address that.
Right. So if it is a biomarker that you're trying to develop, there's a PAR called PAR-19-315 that is set up specifically to discover a biomarker, any type of biomarker. And if it was a response predictor for acupuncture efficacy, for instance, that might fit. But it would have to be, the goal would have to be to develop the biomarker, not for just to be an aside to therapeutic development.
Okay. And Michael, a question for you. What is the difference between the R34 that was issued in March 11th, 2021 and the R61 issued in June?
That is a completely reasonable question. It's an administrative one that deals with NIH's internal rules about how R34s can be used and how R61s can be used. So we originally used the R34 as a planning grant with $500,000 a year. After it was on the street, then they caught it that it didn't follow their established rules. The first receipt date, they allowed it to go through, and we just changed the number on it to R61 and has a new FOA number. But other than that, there are absolutely no difference between the two, except do not use the R34 because it's expired. There are no receipt dates for it. So just the R31. Or 61, R61. Thanks.
Thank you, Michael. Would a novel drug delivery approach, for example, non-invasively delivering drugs to pain foci using focused ultrasounds, be appropriate for these solicitations? And again, I believe the answer to that is no, but you could talk to Nick Langhals about
well, if the technology is already developed, and it's the small molecule that they're caging, that they're developing, would that work, Chuck?
I'm not sure, because like I said, this is really a delivery. So it's really the technology that you're dealing with.
How about contact us and we'll work with you. Write up a specific aims and then we'll work with you.
That's
We'll
one.
find the right
That one.
FOA.
The devil's in the details of that one for sure.
Exactly. Devil's in the details, and we'll work that out.
Is completion of phase 1 clinical trial required end goal and milestone for this RFA? It is a required end goal. It may or may not be a milestone for you in terms of getting to the start of phase 1, surely would be. If you're doing it through our contracts, it'll then be in the contract realm and being funded by them, being funded by NIH Contract world. And then we would, we would be managing that through a contract mechanism. So it's not really a milestone in the grant at that point. Someone asked, does the HEAL program participate in the SBIR program? For these FOAs that we talked about today, there are not SBIR equivalent FOAs for them, but small businesses can apply to them. They just do not have, they're not required to follow the SBIR rules. They're not allowed to do the SBIR profit or fee in our budgets under these things, but you're still eligible to apply to the programs that we have up today.
I'm just going to add on to what Chuck said, and we encourage small businesses to apply to both the R61 and the U19. These monies are a set aside. That's real benefit that you get from the SBIR program, is that it's a set aside, and that money has to be spent for small businesses. That is, we do have a set aside for these particular programs through a congressional mandate for the Helping to End Addiction Long Term, the NIH HEAL initiative. So we encourage small businesses to apply for the R61, the U19s, as well as Chuck's UG3 U19s. They're just not separate SBIR mechanisms.
Correct. And you can always apply through the standard SBIR program as long as you're within the mission of the program, of the institution you're calling to. Should the work that is proposed be done at NIH approved CROs be included in the grant recipient's budget? If you're proposing to utilize our contractors and NIH is doing the contracting for you, then it's not part of your budget and not part of the grant budgets. We work that separately. We do contracting with them separately, and it's completely outside of your budget. Michael, we are at phase 3 of the FDA. We are at phase 3 of the FDA. We're proposing to go to a POC 60 patient test to check the value of pain control, and we are getting from our proposed proof of concept study. So it sounds to me like they're proposing to do a phase 3 study, Mike. So depending on where this could potentially go to things like NeuroNext and beyond, right?
We have if they're really doing a phase 3 study, then we have an efficacy FOA through our division of clinical research. And if it's one of the pain conditions that are in NINDS's mission, the funny announcement would be, give me 1 second, the number will be PAR-21-237. So if you're just doing the clinical trial, it's a phase 3 clinical trial, that's PAR-21-237, but you should really reach out to program staff before you put that application in to make sure that it's at the right stage. Otherwise, we have other funding announcements for it.
Okay. So we are developing a sustained release local analgesic for nerve block to control post-operative pain management targeting 72 to 96 hours to avoid opioid narcotics. Would this qualify? Possibly. It sounds to me like it should. Your proposal would be truly a development project where you're focused on developing the drug formulation, drug product in there. The fly in the ointment could be, are you really moving towards a phase 1 or phase 2 study, depending on what exactly the analgesic is you're utilizing, and is it already approved? And so is this really an ANDA under a new route of administration, or is this truly an IND requiring phase 1 and development? So we would need to talk about the details of it, but I think it's important to discuss that. Again, in a similar ilk, we're developing novel uses for approved generic active ingredients. After filing an IND, we tend to head to phase 2. This is acceptable aim. And again, projects that are aimed towards really just going to phase 2 are not within the scope of the program. So you have to be really focused on optimizing a small molecule or biologic and going into phase 1. So if what you're describing is not that. So there are potentially other mechanisms to do that though. How does the NIH contractor approach work? What can we contract NIH to do? How would we know what NIH is able to do via the contract? Again, talking to us NIH staff would be a helpful first step in that thing. But the NIH contractors in small molecule space can do everything from medicinal chemistry, including synthesis, in vitro ADME, computational chemistry. Our PK tox contracts can do PK in small animals, large animals, toxicology, dose range finding, and GLP tox. Our manufacturing is in place to do, again, small molecules through there. We're putting a contract in place. Hopefully it'll be in place by April, which is probably about a line when these applications would be funded to do biologics manufacturing as well. And the clinical trials can do healthy subjects and potentially patients. But if you have a real specialized patient population, you should talk to us a bit more about what do I think we can get that done within our contracts, or would it make more sense to propose a study yourself? Where would a small molecule that has phase 1 data but needs clinical material development before it could enter a proof of concept trial in Epicnet? I would talk to Barbara Karp at Epicnet about what their capabilities are and what they can do for you. And depending on the mission that you are coming into, I think there's also FOAs in our Division of Clinical Research that might be able to help you in the clinical trial.
If they're SBIR eligible, they could write it as an SBIR.
Absolutely.
The material and then bring it into Epicnet. That's a.
But I believe Epicnet is aware of this issue and discussing how they might be able to help with it too. So again, I would talk to Barbara Karp. Is phase 1 study being conducted in the U.S. or can it be conducted outside the U.S. as well? If they're done through our contracts, they're done in the U.S. That's where our vendor has sites for the most part, although PPD, I think, does have some worldwide sites. Could it be done outside the U.S.? In theory, it could, especially if you were doing it yourself. But you would need to justify why that would be necessary. And there's some concern, obviously, from the NIH side of losing control of what is happening in patient protections when the trials are done outside the U.S. Should work that is proposed to be done at NIH approved CROs be included in the grant recipient's budget? I answer that question, no. If it's NIH CROs, then not part of your budget. Are THC and CBD used for pain management qualified for this program? Again, the devil's in the details. What are you doing? How are you optimizing them? I would need more details to answer that question. And we're happy to set up calls and talk to you in detail, especially if you don't want to talk about some stuff that may be more intellectual property protected, because we can have private conversations where that's not an issue.
That's all I have so far, Chuck.
Okay. Any other last minute questions? You can get them in. I will be making the minor corrections on those slides to say R61, and I will have Rakanda send out a PDF of these slides that I used today, later today, hopefully. And we're happy to set up phone calls. Myself, Marianne, Michael, Anne Marie will all take calls based on what your questions, relating to. But feel free to reach out to me directly, and I can forward them to the appropriate people, or you can reach out directly if you know who you want to talk to that person. And the most important thing is get a set of specific aims together that says, here's what I want to do, 1 to 2 pages. If you have those with your request, it makes the request a lot easier to get the right people on the phone call with you. Do appreciate everyone's time. I look forward to hopefully seeing your applications in the future. And if you have any follow-ups up, feel free to reach out via email, and let me unpause my thing and put up the email. Resume. These are our emails, and I'm sorry, Marianne, I didn't put your email up here, but is it M. Pellimonter or Mary? How is your email?
It's Mary.Pellimonter
Mary.
at NIH. Yep.
There we go. So it follows the same thing, Mary.Pellimonter.
Yep.
Okay. Perfect. So if you have any questions, or feel free to reach out to any of us, and we'll be happy to answer them. Thank you again for taking the time. Look forward to seeing your applications in the future. Enjoy the rest of your day. Thank you, Michael.
Thanks, Chuck.
Thank you, Marianne. Thank you, Rakanda. Great job.
Welcome.